Recall in mitosis that we had the splitting of the cell into two identical daughter cells. Each of those identical daughter cells had the same number of chromosomes in each. So the cell first copies the chromosomes, then it's going to divide through the different stages of mitosis. So the daughter cells are identical to the original. So if the parent cell has 46 chromosomes, each daughter cell would also have 46 chromosomes. Or if the parent cell had 20 chromosomes, each daughter cell would have 20 chromosomes. It would always be identical. Our chromosomes are diploid, which means they have two copies of each. So we have pairs of chromosomes. And the human's diploid number is 46. We have 23 pairs of chromosomes for a total of 46. The haploid number of that is 23. Now, that's great at mitosis, making all of these identical daughter cells. It's great for repair if you get cut, if you get broken bone, for growth. However, in sexual reproduction, when the sperm and the egg combine their chromosomes, 46 and 46, we don't want it to equal 92. We need to get back to that 46 number. So before we can make the sperm and the egg, we have to half the number of chromosomes. We need to get to that haploid number, so that when a sperm has 23 chromosomes and an egg has 23 chromosomes, when they come together, we get that complete 46 number of chromosomes. So kind of just a little recall, parents are diploid. They have the full set of chromosomes, the pairs of chromosomes. But in meiosis, our new process, not mitosis, but meiosis, we're going to try to get to that half number. We need to half that number of chromosomes. So when the gametes, that's the sperm and the egg, with each half the number of chromosomes through fertilization, they're going to form that diploid number. They're going to form the zygote. And it's that zygote that can then go through mitosis, and it becomes the next diploid generation. So those numbers of 46 chromosomes keep reproducing, and each new cell is going to have that full 46 number. So in meiosis, we are getting to that half number. So mitosis was just making identical daughter cells. Meiosis, when you're looking at halving that number of chromosomes, just specifically so we can have half the number for sexual reproduction. So this picture I had on the last slide, or a similar one to that, just showing that even in humans, we have 23 pairs of chromosomes. We have a diploid number. Everything's in pairs. So we've got 23 different chromosomes, and we've got a duplicate copy of each. Now, the fact that we have a pair of each set of chromosomes, we call these chromosomes homologous. That homo meaning the same. And when I'm talking the same, I'm saying that each chromosome carries the same genes for a trait. So let's just say, for example, our chromosome 1, whether you're the chromosome on the left or the chromosome on the right, because you got pairs, that this little green band could control the trait for hair color. And this would also control the trait for hair color. So the trait for hair color could be, it's not, and it's spread over multiple chromosomes, but it could be found just on chromosome 1 in this one same location. So they have the same length. They're even grouped and separated out by length. And one of them came from mom, so the information on your hair color, some information came from mom, some information came from dad. The information that controls the different traits, such as hair color, is found in the same location on those chromosomes. So these chromosomes are carrying the same kind of information, hair color, or maybe they have eye color or height, or some other feature. There might be some differences, yes, but they're controlling that same type of information. And so they are identical in the type of information that they carry. They're similar in shape. They're similar in location of where that information is. They're similar in size. So they're very similar to each other. Their centromeres, where eventually they will connect with each other, are also going to be in the same places. And yes, when they're stained, they show similar banding patterns, that you can see the different colors based on the different bands. So these homologous chromosomes, they do have genes that control the same trait, such as hair color. They have lots of difference, though, they have lots of gene variants. The variants are called alleles. So let me give you an example, because here's some terms, and these terms are going to come up in our next unit. So it's a little bit of preview. So a gene is a trait that controls something, such as the color of your eyes, the color of your hair. The different forms of the genes are called alleles. So for an example, you have a gene for eye color, but the alleles, these are the different forms of the gene. You have alleles for blue eye color, brown eye color, hazel eye color, and every other shade in between. So you have a gene that controls a trait, but then you have alleles, which are like the differences amongst those traits. Because those alleles and genes are going to come up in our next unit when we start talking about, do you have the same information, which is known as homozygous, or heterozygous for those alleles. If your alleles are the same, meaning mom gave you the information for brown eyes and dad gave you the information for brown eyes, so the only information you have for the color of your eyes is brown, that's homozygous. You have the same information on both of those homologous chromosomes. If, however, different, like dad gave you dad gave you brown eye color information and mom gave you blue eye color information, because those alleles are different, we call them heterozygous. So you still have information for eye color, and then our next unit we'll talk a little bit more about, so what eye color would you have? Which one wins out or beats out? So I'm going to bring up some of this. So how do the sex cells replicate? So just a little bit of overview of meiosis compared to mitosis. So meiosis is broken down into two phases. We have Meiosis I and we have Meiosis II. That in itself is different than mitosis. Mitosis, we just had mitosis. It had different phases of prophase, interphase, anaphase, I was going to say it wrong, prophase, metaphase, anaphase, and telophase. But meiosis is going to have those too, but it has two distinct sections or divisions. So we have Meiosis I, that's going to go through the pro, meta, ana, telophase, and we're going to have Meiosis II, that's going to go through the prophase, metaphase, anaphase, telophase. So the differences between Meiosis I and Meiosis II, and this is the key difference that if you're taking notes, you should be writing this down. Meiosis I is where it halves the number of chromosomes. So if we started off with 4 chromosomes at the beginning of Meiosis I, the cells at the end of Meiosis I will have 2 chromosomes. So it halves the number of chromosomes. So you're like, well, what's the point of Meiosis II then? Well, Meiosis II still undergoes another cell division. It still goes through that prophase, metaphase, telophase, anaphase, and it's where the chromosomes are going to separate. So at the end of Meiosis I, we're going to have 2 daughter cells. We started with 1, now we have 2 daughter cells, and they each have half the number of chromosomes. Meiosis II, we're going to take those 2 daughter cells, they're going to divide again, and they're each just going to have a single copy of the chromosomes. So just a couple things. I know this is a little previewing, and I've got lots of pictures to show you this coming up. So the chromosomes are still going to get replicated prior to meiosis. That's just what's going to happen during interphase. You're going to have 2 sister chromatids. The big difference here is that the chromosomes, those things that look like X's, because we've got double the information now, the X's are going to pair up with X's. So you're going to always have a pair of X's. That's different than mitosis. Mitosis, we had X's, and they lined up in single file during metaphase, but they didn't pair up with other X's. So in Meiosis I, those X's, those chromosomes are going to pair up with each other. Once those chromosomes pair up, we call them homologous pairs. The homologous is because they're going to pair up with the similar sized chromosomes as each other. They're going to find other ones that are the same size that are carrying the same type of information, such as hair color, eye color. And it's those that are going to separate apart. Meiosis II, there is no interphase between Meiosis I and Meiosis II. We're not going to duplicate anything. Again, our purpose was to half the numbers, so we want to stay halved. So all that's going to happen is those X's, which haven't divided into 2 yet, they're still an X, are now going to separate. And Meiosis II is going to look very similar to metaphase, where those X's get yanked in half and the sister chromatids have to say goodbye to each other. So we're going to take a look at some of these pictures. So just remember, in meiosis, we have a meiosis I and a meiosis II. Meiosis I is where we get half the number of chromosomes. Meiosis II, we just have another cell division. So now we're going to have 4 daughter cells at the end. So this is showing at the very, very beginning. Our original number of chromosomes is we have 4. That's our diploid number. We have 2 sets. We've got 2 blue. We've got 2 red. We have a total of 4 chromosomes. As we go into meiosis I, if we notice here, this is when we've duplicated. They're not a single strand anymore. They've doubled their information, so now they're X's. But the unique thing about it is they've paired up. X's pair up with X's, and they do pair up with a similar size, similar information. So at the end of meiosis I, all we've done is that we've taken 2 X's, they've gone to one cell, and 2 X's went to the other cell. And so if you notice, we only have 2 chromosomes here. Yes, it's duplicated information, but we only have 2 chromosomes. And the other one only has 2 chromosomes. So this is where we went from 4 chromosomes to 2 chromosomes. So at the end of meiosis I, we've half the number of chromosomes. But we don't want those X's to stay together. That's when we go into meiosis II and we yank apart the X's. And so this red X is going to get divided into 2 and the 2 new cells, and this blue X is going to get divided into 2 and go into these cells. So we end up with 2 cells at the end of meiosis I, and they have half the number of chromosomes. At the end of meiosis II, we have 4 cells, and they still have that same number of chromosomes. It's just not duplicated anymore. Now, interesting part about meiosis is that this is where we get a lot of genetic variation. And it's why you may or may not look like your siblings, if you have any siblings, and why you may or may not look like more like your mom or more like your dad, is because of all the genetic variation that can occur during meiosis. And one area where we can get some different variation is during a lot of the prophase 1. This is what's happening during meiosis I, is that when these chromosomes, these X's pair up, when these X's pair up, because they contain similar information such as eye color or hair color, they can easily flip. They're so close together, they can just exchange a little bit of information between there. And so they truly are doing that, is they exchange some information, so a little bit of this information goes on the other chromosome and they just flip-flop. So now we've got chromosomes that aren't identical to the original parents. They are still going to contain the same type of information. They're still going to contain information for hair color, eye color, whatever. But it's slightly different than the original because we've got a little mix-up or crossing over here. And they can easily do this. So these 2 chromosomes are held together. There are different lattice work that holds them together, and they contain similar information, but they can easily, it's called crossing over, because as they cross, they get cut and realign themselves or reattach themselves. So this is where we get quite a bit of genetic variation with all of this crossing over. And it doesn't have to happen in just one spot. This can easily cross over and cross over. This is showing where you've got a couple crossovers. And so this one blue strand didn't really do anything and it stayed the same, where some of these other strands did a whole bunch of mixing up. And so we've got this huge bunch of genetic variation that's occurring. Another way we can get some more genetic variation is what's known as independent assortment. So when those homologous chromosomes, those paired chromosomes, align themselves up during metaphase, so they align themselves up in the middle before they get yanked apart, it's random in the order that they line up. So this is showing where all the mom chromosomes are on one side, all the dad chromosomes are on the other. So when they get yanked apart, all the red go to one cell, all the blue go to another cell. But that's just one combination. This is showing where 2 red ones and a blue one over here, and then it's the reverse. So this cell's going to have 2 reds and a blue. The blue's going to be the big one. This one's going to have a blue, a red, and a blue, and depends on which blues or which reds they get. Now, this is just showing there's 8 different assortments in which direction and how they can line up. But we have 23 pairs of chromosomes. We have 46 chromosomes. That's a huge number of variety in all the different ways that they can line up. Now, this also might be more an example of if you look just like your mom or you look just like your dad, you probably got a whole bunch of chromosomes from your dad, or you got a whole bunch of chromosomes from your mom. Kind of, again, just how they paired up. If you look a little bit more like one sibling compared to another, maybe that random assortment on, how they lined up was very similar for you compared to other siblings. If you look nothing like your siblings, just means you guys got completely different groups of chromosomes from your parents. It's a lot of randomness. And the fact that it's completely random which sperm and which egg meet to mix all of this genetic variation is also random. So just looking at numbers alone and how random this can be and why we have so much variety. So there's, based on the number of chromosomes that are donated by the parents, again, you have 23 pairs of chromosomes. So there are, in theory, 70, I was going to say trillion different over 70 trillion different chromosomal different zygotes that are possible. And if you have crossing over that occurs just once, we're talking even more astronomical different zygotes that are possible. So this is why you do not look exactly like your sibling unless you're an identical twin. The chances of that ever happening, literally 1 in quadrillion. I'm not sure what number that is at that point. So, and usually this is only looking at numbers if crossing over occurs only once, but crossing over usually occurs several times in each chromosome. So this is why we don't have siblings looking identical, is because there's a lot of variation that can happen with that crossing over and how they line up, and then which sperm meets which egg. But why do we care about genetic variation? Because in mitosis, all the cells are identical to each other. Asexual reproduction, mitosis, identical clones. Now, that is less advantageous if in a changing environment. So if the environment is changing, we don't want identical clones to each other. It means we don't have anything different from each other to be able to adapt to our environment. So sexual reproduction through meiosis causes a genetic recombination, again, trillions of different outcomes among members of a population. And this is going to be advantageous in a changing environment. And we're going to get into this in a couple weeks when we start looking at natural selection. Just the fact that we can have so many differences in the same species. And we're, that's looking at some butterfly wings and just the genetic variation they can have, but just the genetic variation even among humans themselves or any animal out there. If the environment changes, because you can have so many differences, it's more likely that one of these differences is going to be advantageous. It blends in better with the environment and hides from some type of predator. It's going to be an advantageous difference. And so whatever organism it is survives. It increases the likelihood of survival. If everything looks identical to each other and you have one small change, maybe the change in color and now something doesn't blend anymore, you could wipe out an entire species. So it's good to have genetic variation. Now, just looking a little bit, meiosis, kind of the main points of what's happening at each of these phases. So in meiosis I, again, just to recall, we do half the number of chromosomes. So we still have prophase, metaphase, anaphase, and telophase. Just to distinguish them as different between meiosis I and meiosis II, we call it prophase I, metaphase I, anaphase I, telophase I, just so we know we're in metaphase, so we know that we're in meiosis I. So in prophase I, it's really the same initial events that happens in mitosis prophase. You're still going to have spindles getting formed, you're going to have the nuclear envelope that's going to disappear, you're going to have sister chromatids that are happening, but you're also going to have the pairing up of the homologous chromosomes. So that's going to be a difference. So we start to get, we call them tetrads or homologous chromosomes because the pairs of chromosomes pair up. In metaphase I, we are still meeting along the middle. The only difference is we don't meet single file, we meet in pairs because we have homologous chromosomes in metaphase I. In anaphase I, it's still a sad phase because they're still getting yanked apart, However, the whole chromosome itself stays together. So you have one full chromosome that is saying goodbye to its other full chromosome. So the pairs split in half, and this is where really we have our number. So in mitosis, what separates is the actual chromosome. The X yanks apart. And here, we're just separating the one X from the other X. So one full X goes to one side and the other full X goes to the other side. But it still stays as an X. It still stays as that X chromosome. So the sister chromatids do not separate yet. In telophase 1, we have, again, still a little bit of reverse of prophase 1. We're going to have that nuclear envelope reappearing. We can have a nucleoli reappearing, and we can start to see that we're going to start to separate and become two cells. My only difference now is each new cell has two chromosomes, where initially it would have had 4, so we've halved the number. Now, before we can go into meiosis II, we have what's known as interkinesis. Inter just means between. It's not a full interphase because we're not trying to duplicate anything. We're not trying to make any new DNA. We need to keep that DNA number in half. We do need to duplicate some chromosomes, or not some chromosomes, excuse me. We do need to duplicate some organelles, but we are not duplicating the DNA. So it's a shorter time period before we go into meiosis II. So we're going to take those two new cells that we just got, and each one has just 2 chromosomes in it, although they are duplicated chromosomes, so they're shown as an X. And we're still going to go through prophase, metaphase, anaphase, and telophase. Why I don't have separate slides for each of these is these should look pretty similar to what happens in mitosis. So in prophase, you're going to see the nuclear envelope disappear. You're going to start to see those spindle fibers going out. Those spindle fibers will line those chromosomes single file in metaphase 2. They're going to get yanked apart in anaphase 2. Telophase, we're going to get that nuclear envelope reappearing, and ultimately that cell is going to divide into half and we have 2 new cells. So this looks pretty similar to mitosis. So it's kind of the exact same thing happening. We're just dividing those duplicated chromosomes into half. My recall, I just want to go through meiosis I. We halved the number. We had 4 chromosomes. They paired up. That's unique. They pair up, and then the pairs of chromosomes, the X's, say goodbye to each other. So at the end, we have half the number of chromosomes, but those X's have to get divided in half, and that's what's happening in meiosis II. So at the end of meiosis II, we have 4 cells, and each cell has just 2 chromosomes. It has the haploid number. Now, little video showing this occurring.
Meiosis has 2 major divisions, which lead to 4 cells that have a haploid number of chromosomes. It starts with the diploid cell, in interphase, where DNA replication will occur. Then meiosis I can begin, splitting the cell into 2. The cells don't replicate their DNA before the next division. In meiosis II, the chromatids separate into chromosomes, leaving 4 haploid cells. These haploid cells will mature into gametes, sperm, or egg. Let's look at this process step by step in more detail. During interphase, the DNA is replicated in preparation for meiosis I. The chromosomes aren't actually visible yet, but I'm showing them as chromosomes here so that it's easier to see that we'll follow a pair of homologous chromosomes through the process. The first step of meiosis I is prophase I. During prophase I, the nuclear membrane dissolves, and the centrioles move to the poles. But something very strange happens with the homologous pairs of chromosomes. The duplicated homologous chromosomes are called a tetrad. And when the tetrad pairs up, it's called synapsis. Once they're paired up, they will recombine their genes by crossing over. The duplicated chromosomes are no longer identical, which creates variation and diversity in the final gametes. Spindle fibers will attach to the chromosomes. and they'll move and line up in the center during metaphase. Now, which side they face is random and is also known as independent assortment. They will not be pulled apart at the centromere like in mitosis, but instead, homologous chromosomes will separate. At telophase 1, the nuclei reform, and the chromosomes wind into chromatin. And during cytokinesis, the rest of the cytoplasm divides. In humans, there's no second interphase. However, some plant species experience a second interphase, or interkinesis, but the DNA is not duplicated in this phase. During prophase 2, the chromosomes reform and the two nuclei break apart. Then the centrioles send out spindle fibers and connect to the centromeres of the chromosomes. During metaphase, the chromosomes line up at the center. This time, it's in the same direction as it would be in mitosis. Then in anaphase 2, the chromatids separate into chromosomes as spindle fibers pull them to each side. In telophase, the chromosomes unwind into chromatin and the nuclei reform. Then the cytoplasm divides in cytokinesis, and we're left with 4 haploid cells that are all different. Again, those differences are because of crossing over and recombination. Let's look at the process of crossing over one more time. These duplicated chromosomes are homologous. One came from the mother, and the other came from the father, but they both carry the same type of information. During synapsis, sections of the chromosomes will cross and switch, creating recombinant chromosomes.
So, just doing a little comparison back and forth on what's happening in meiosis compared
Thanks for
to
watching
what
this episode of
happens in
Teacher's
mitosis.
Pet. Don't forget to like and subscribe
so in
and follow.
meiosis, you have 2 divisions. You have that Meiosis I and Meiosis II. Mitosis, you just have one nuclear division. There is no mitosis 1 or mitosis 2. In meiosis, the chromosomes are going to synapse, they're going to pair up together, and you can have that crossing over occur. Where in mitosis, none of that happens, no pairing up of chromosomes. Meiosis, the centromeres survive anaphase 1, meaning they hold the 2 halves of that chromosome together. They stay as an X throughout anaphase 1. In mitosis, they yank apart. The sister chromatids say goodbye to each other. Meiosis, you have half the number of chromosomes. Mitosis keeps the same number. Meiosis, you have 4 daughter cells in the end. Mitosis, you just have 2 daughter cells. Meiosis produces daughter cells genetically different from the parents because of that crossing over and how they line up. Mitosis, you have daughter cells that are genetically identical to the parents and to each other. Meiosis is used only for sexual reproduction, whereas mitosis is used for both asexual reproduction, such as in lots of different organisms like bacteria, and it's also used for growth as well, growth and repair. Now, individuals are diploid. We do have the duplicate set of cells. However, we do produce haploid gametes, meaning we produce eggs and sperm that have half the numbers. So the only haploid part of the life cycle are those eggs and sperm. So the products of meiosis are always eggs and sperm, nothing else. So meiosis occurs during the making of gametes. It's known as gametogenesis. Genesis means to make. The gameto means the gametes, meaning the sperm and the eggs. So when sperm are made, when you get those 4 final cells at the end of meiosis, all 4 of those become sperm. What's unique though is that eggs, all 4 of you still get 4 cells at the end. Only one of them actually becomes a viable egg. So you get 1, it becomes a viable egg, and you get 3 that we call as polar bodies. So in the human life cycle, sperm and egg are produced by meiosis so that they have half the number of chromosomes. They fuse at fertilization and they result in a diploid zygote. And then that diploid zygote then starts to undergo mitosis. It begins to grow into a full-sized adult with enough mitosis and enough years. So this is just showing that in adults we are diploid. We have the duplicate set of chromosomes. But in meiosis, the making of the sperm and the egg, it has half the number. So instead of having a full 46 chromosomes, each one only has 23 chromosomes. During fertilization, all of those chromosomes meet up. This, instead of drawing all the 23 in each of these, they just drew 3. and they have the full number, that 6, or in real life 46, and those cells then begin to divide through mitosis, so that every other cell after that is going to have that diploid number. But what happens if there is an inappropriate number of chromosomes? What if there's a mix-up somewhere in all of this alignment and dividing? So if the correct number of chromosomes in a species is present, we call it euploidy. However, if there's a change, we call it aneuploidy. So this can happen a lot when they start pairing up and dividing. It's called non-disjunction. So we can end up with only one particular type of chromosome, and so we should have pairs of chromosomes. But what if you're missing one side of that pair? We can have monosomy. Trisomy is if you have an extra chromosome of one of those. Now, how that can happen? So what is this non-disjunction? Well, this is showing if non-disjunction happens in meiosis I. It's an unequal separation. So in meiosis I, we have 2 chromosomes, and at the end of meiosis I, each new cell should have half that. It should have one of these X's in each one. But what if they didn't split? What if the 2 chromosomes went to one side and the other cell didn't get any chromosome? And then it followed it through meiosis II. Well, these eggs still have nothing. You can't get anything from nothing. They still go through meiosis II, and the X is split into half. But each of these 4 eggs should have 1 chromosome. Instead, 2 of them have 2 chromosomes, and 2 of them don't have any chromosome. And then through fertilization, the sperm all still have the correct 1 chromosome, but all of a sudden we can end up with 2 eggs that just have 1 chromosome, and we can have 2 eggs that have 3 chromosomes. So instead of having 4 eggs, each with 2, we have 2 with 3, and we have 2 with only 1. And so we can end up with 4 chromosomes, or 4 fertilized zygotes that are all mixed up. 2 of them have monosomy and 2 of them have trisomy. If non-disjunction happens a little bit later in meiosis II, so this is showing after meiosis I, we've halved the number of chromosomes. That's what this should look like over here. We've halved the number of chromosomes. However, what if the X doesn't separate? What if those spindles don't yank the X's away from each other during meiosis II? If it's going to happen normally, you have the normal number, 2 of them, the X gets yanked in half, they each have 1, they fertilize with that sperm, and each of the resulting zygotes has the correct number. But if this doesn't separate in half, and both of those halves go to one egg, and the other egg doesn't end up with anything, we're right back to where one zygote has monosomy, and another zygote can have trisomy. And so this happens, more frequently than you would think. Depending on which chromosomes don't get separated out equally, can mean that the fetus, that zygote, cannot develop. It cannot go through mitosis. There is a checkpoint in there, and it wouldn't develop normally. However, depending on which chromosome it is, it can still, that infant, that baby kid, that zygote, whatever you want to call it, can still grow normally. And the most common type of this non-disjunction that happens is when you have a trisomy of chromosome number 21. And that's when we call it as Down syndrome. A lot of the other chromosomes, if you have extra ones or are missing ones, the fetus just can't survive. But with having an extra of the 21, you can survive. And it's known as Down syndrome. Just some characteristics of Down syndrome individuals. They're usually a little shorter in stature. They usually have an extra eyelid fold. Their face is a little flatter. Their fingers are a little shorter. They usually have a wider gap between their first and second toes. So just kind of some common characteristics among those that have that extra chromosome. Now, things that can increase the trisomy or the monosomy that can increase the chance of this non-disjunction, a lot of it's the age, and usually it's the age in women, that males can make sperm throughout their entire life, and there's usually not a lot of this non-disjunction that happens during the sperm. It can, but it usually seems to happen in females making eggs, and the eggs don't divide those chromosomes equally. So usually when you start to hit women over the age of 35, the chances of those non-disjunction happening increases. But I was going to say, it's just age. It increases your chance. It's definitely not a must that it would happen. They just look in statistically that chance of kind of that mix up and not catching it increases as both the females and males do get older.